And we are live. This is Salvador Cordova, and I'm privileged to be joined by Professor of Chemistry, Associate Professor of Chemistry, Dr. James Carter, and we will be talking on a nerd topic. Fasten your seatbelts. Anytime it's me and Dr. Carter, it's usually going to be pretty technical. Um, we have a spectrum of things that we discuss on the channel. And there is, when we start talking creationism, which constitutes about 65% of this channel, it can get pretty technical because the audience that I've been trained and that I have a heart for are college students, college graduate students, professors, and scientists. So there's a lot of the content on the channel that is targeted to that audience, and this is going to be one of those shows. I have been hopefully like the last two nights just trying to balance out the channel with things more accessible. We were doing scripture readings, which meant a lot to me. We were commenting on culture, things that I think every churchgoer, every Christian can relate to. And so some of the, tonight is definitely a nerd night. I should mention that to kind of alleviate the nerdiness of the channel, there is a sister channel called the Evidence Reasons Academy. If you go to the About page, you can click there, and you can go there. I'm planning to be doing chemistry lectures now that my schedule's a little freer now. And I also need to do the lectures so I can relearn basic chemistry. So I'm just starting with general chemistry. And I probably will be trying to work my way to organic. I've had biochemistry, but I could always review that. And there's also molecular biology. So this is really great to be able to meet practicing scientists. And Dr. Carter, could you introduce yourself? You're muted, Dr. Carter.
Oh, there we go. Yeah, I had to clear my throat, so I muted. Yeah, some of you already know me. I've been a teacher and an associate professor, taught 15 different science courses over the last decade. Thinking of switching gears now. I realize that I like learning so much, but repeating myself over and over is getting kind of tedious. So I kind of like this format of YouTube where it's like you explain something, and then people can keep watching it anytime they want. They can watch it at any speed they want, whenever it's convenient for them. So this asynchronous learning seems to appeal to especially the younger generation that's grown up with the internet and YouTube. So yeah, I found Sal's channel. I don't know, maybe has it been a year already, or maybe not that long?
I think it was like last November we started to talk and get introduced.
Yeah, so it's about half a year.
Yeah.
So yeah, it's just been a real privilege. I really like coming on Sal's channel because you don't have all the distractions and interruptions of people who would like to thwart our efforts. Sal and I have similar dispositions and hearts, in that we, our atheist friends, our brothers, we love them, but like siblings, we don't want them killing one another. So on the one hand, we love them. On the other hand, we don't want them taking anyone down. In our viewpoint, in our opinion, the work that they're doing can be counterproductive and destructive, and I think they feel the same way about us, that we're kind of on opposite ends of a team that are battling it out for the minds of people and trying to make the world a better place. And so it's the worldviews are kind of head to head right now, whether there's a all-powerful, loving God and what that God is like, or if things had to come about by a random chance. And that's what we're going to look at tonight, is what are those chances of life coming about through just without any intelligent input, just through the interaction of chemicals, through physical and chemical means and selective processes. So that's what we're going to look at tonight. It's kind of a short paper, but a little heavy on the math. So I asked Sal to help me out with that. We don't need to go into the advanced math, but you'll get the gist of what the paper is talking about really quickly here. So if there's anything else you wanted to talk about before we get started, I can do a summary and kind of an overview, but while we wait for more people to trickle in here.
Yes. So maybe just as filler, I found out a few things that may be of interest to you. It is also the improbability of origin of life, but this is for, well, actually it is relevant because these are nucleotides. So this is one level lower. And it's been a while since we've actually had a live stream together. And there was actually one time we were interacting in the side chat. And I tell you, I spent half a night digging up references, obscure. Unfortunately now, university privileges I've had for like 17 years have been suspended. They may have just been cleaning up the register because I graduated almost 17 years ago. No, the last time I was a student at George Mason University was about 2004, and I've had library privileges for the last 17 years. They may have updated their system and just realized, hey, what's this guy doing here? You know, So I mean, it's not like they totally deplatformed me or anything. It could have been. I was just actually more amazed that they had granted me privileges for so long, but. It just so happened maybe the last month I had privileges, I was able to dig up these papers. So let me show them to you. And to those in the side chat that haven't had chemistry in a long time or ever, if you want us to slow down and explain terms or something, please say something, tag me. I try to watch the side chat, but when I'm also running all the controls on the channel and running a show, I can miss things. So please, I plead your forbearance, but I am interested in answering questions, especially since Dr. Carter is here who can help me answer the questions. So the last time we talked on the side chat was maybe 3 months ago. And I was like, we were talking about this particular sugar and I learned a few things. I had to, let me share this a little bit with the audience. Learning a little bit about organic chemistry involves staring at diagrams, and I could stare at diagrams for a very long time before I begin to get. So you can imagine me just looking at this diagram for an hour or more. I mean, maybe I'm just slow, but that's what it takes for me to kind of digest. And then everyone can draw their diagrams a little differently. And so that can be a little disorienting, but then I'm trying to, one way that I learn the diagrams to try to draw them. So if I look at it and then I'll go somewhere and draw it and say, okay, this is what's this. So, I know you recognize this one here, Dr. Carter. This is the D2-deoxyribose, the furanose. It's beta. It's a beta form. And we normally just call it deoxyribose, but really there's so many of them. So when people talk about DNA and deoxyribonucleic acid, that's actually not the most complete name, but if we go by all the the most rigorous nomenclature, it can get pretty tedious. So
yes, it's definitely a mouthful to try to even pronounce all of these things, let alone say them. So we do try to go for shortcuts. And now I'll just point out a few quick things about the sugars. They're otherwise called carbohydrates because they're hydrates of carbon. So if you look at, this is called a Fisher projection, and just to save time and make it easier to understand the three-dimensional orientation of whether this hydroxyl group, this OH, is on the right or on the left, we draw them in this vertical position where there's a carbon everywhere you see an intersection here. So this would be a 5-carbon sugar. So we call that a pentose, like a pentagon has 5 carbons, and ribose happens to be a 5-carbon sugar. A lot of the sugars are either 5 or 6 carbons. And if they're at least 3 carbons long, they can form these ring structures, and the way they're drawn here, they're called Hayworth projections. And so if you imagine this is a flat ring that's kind of, tilted back, so it's this fatter edge that's towards you, and then this oxygen is further away from you. And you might be wondering, what are all these numbers here? Well, we like to number the carbons to help us orient, okay, what's the orientation of the hydroxyl group at position number 3 or number 4? And that apostrophe is just because there's another set of carbons that you find in the bases themselves, like the adenine, guanine, cytosine, thymine, uracil. They have their own carbons, and so we have a separate numbering system for them. So they just get 1, 2, 3, so on. And then the sugars get these apostrophes, so we call those prime, 1 prime, 2 prime, 3 prime. And the reason why it gets the name deoxy is because on carbon number 2 prime here, it's missing an oxygen. It just has 2 hydrogens, which aren't shown. Every carbon likes to have 4 bonds. So right here is a carbon. There's one.
Here's maybe a little bit more elaborate.
There you go.
Yeah.
Yeah, there's
easier.
ribose, which you find in the sugar in RNA. There's deoxyribose, which you find in DNA. So DNA stands for deoxyribonucleic acid. When you take the sugar and you attach a base over here, and you attach a phosphate over here, you have a nucleotide. So it gets a little bit tricky here. The base is the adenine, guanine, cytosine, thymine. When you attach a sugar to that base, you change the name a little bit. It becomes a nucleoside. Think S for sugar, nucleoside. And then adenine becomes adenosine, guanine becomes guanosine, thymine becomes thymidine, cytosine becomes cytidine, and uracil becomes uridine when you've got a sugar attached. And you can also attach 1, 2, or 3 phosphates, and then it becomes a monophosphate, a diphosphate, or a triphosphate. So what you find in your DNA is deoxyribonucleic acid. So when they start linking together, they become nucleic acids. So yeah, the difference between DNA and RNA, the D stands for deoxy. It's missing this hydroxyl group, like over here. And can you see my cursor, or is it? Do I need to share my screen to do that?
Oh, absolutely not seeing your cursor.
Yeah, okay, that's not helping. Yeah, you're doing good though. You're pointing over the right section. If you could just show that pink hydroxyl group.
Okay, let me see.
On carbon number 2 prime.
Let me see if I can. I'm sorry. I also had my set of technical errors here.
Oh, there we go.
So this
is there we go.
So, by the way, just for the reader's benefit, a hydroxy group is spelled OH or H. Sometimes you'll see it HO, but the O is connected to the C. So if you just see, okay. So that's just for the readers that if they want to know what a hydroxy group is.
That's right. Yeah, we don't want to. We don't have time to go into explanation why carbon likes 4 bonds and why oxygen likes 2 and hydrogen likes 1. But there is a bond, a covalent bond, that means they're sharing electrons between the carbon and the hydrogen. There's a single bond there. So when you see lines, that means they're sharing electrons. That's a covalent bond because they're sharing their valence electrons. And so ribose is the one that you see in RNA, ribonucleic acid, and deoxyribose is the one you see in DNA. Now, if you go to let's see, do you have a picture with the base attached as well as the?
No, not in this set.
Okay, that's fine.
But now this is be a little frustrating when you. I'm going to show. It's too bad I can't show them side by side, but you see how it's rendered here for the viewers, and I was pointing out that all the diagrams can be. It's almost like handwriting. Some people will just. It'll look a little different, and sometimes there's a little bit of shorthand, and it doesn't look exactly the same. You're like, is that really, are we talking about the same chemical? And that's something that I found that I had to work through. So
Yeah, if
this
you stay on there for a second, those are all the same molecule, but about 99% of the time, at least for the 5-carbon and 6-carbon sugars in water, they will do a self-chemical reaction where they form a ring structure. And that's what you're seeing above and below. So in the middle is how you would see it about 1% of the time where it opens up and that Fisher projection. And then there's a chemical reaction that will take place where it will fold spontaneously into a ring. And that's what you see down below there, that the Hayward structure, the hexagon shaped structure. But it can actually come together in a couple of different ways. And of course, only one of those is the way you find it in nature, but that means there's a lot of other ways that can go wrong. And for instance, those hydroxyl groups, like we see in the middle, they're pointing to the right. If just one of those is pointing to the left, it wouldn't be ribose anymore, or deoxyribose. It would be a completely different sugar with different properties.
Like this
deoxy, like you see there on the left.
Xylylose.
That's right. Xylose is a sugar found in trees and berries and stuff. But it's you can see the difference between it's the same number of carbons and hydrogens and oxygens, so they're isomers of each other, but they're arranged in 3-dimensional space differently. Whether that OH group is pointing on the left or on the right will change which sugar it is. So, and
in fact, the chemical formula would be C5H10O4. Yeah, no, is it O5? Because we should be riboperin. Okay.
Well, if it's deoxy, it's going to be O4.
Okay, that's what I meant. Yeah.
Yeah.
So, what Dr. Carter is mentioning, these all have the same chemical formula, like this C5H10O4 for deoxy, and it's O5 for just regular ribose, but or something like that. But you would actually have different names. So I'm just confusing things. The point is all of the molecules you see here actually have the same proportions of hydrogen, carbon, and oxygen, and they therefore have the same chemical formula. What do you call this kind of formula when it's just with the numbers?
That's the empirical formula. So like if you were just to, run it through, some experiments that would just figure out the mass of carbons and the number of carbons and the number of hydrogens, the number of oxygens, then you're empirically determining just strictly how many atoms there are, but it doesn't tell you how they're arranged in 3-dimensional space. So what you see below that is just a handful of the different ways that you can arrange 5 carbons, 10 hydrogens, and 5 oxygens. There's basically an H2O for every carbon. That's why it's called a carbohydrate, because you're basically attaching water and H2O to each carbon. But one of the hydrogens will go on one side, and then the OH will go on the other side. But if you look at all those structures that you had below on that previous slide.
On this one,
yeah, you can see how if you pay attention, they kind of look similar, but they're not. Like in one case, they're pentagon shaped, and in another case, they're hexagon shaped. But if you count the number of carbons at each of those intersections, it's the same. It's still 5 carbons. These are all different pentoses, 5 carbon sugars. And then the D tells you, well, you can't tell very much from whether it's the left-handed or right-handed isomer from these Haworth projections, but in nature, you almost always find the sugars in the right-handed configuration. That D stands for dextrorotary. It just means that it rotates plane polarized light to the right. Don't worry about it. But if you're wondering what that D means, and then the L, just think of left-handed, but it really stands for levorotary or levorotary. So left-handed and right-handed. There you go. You can see, well, you can't really tell here. Yeah, I guess you can. The L versus the normal D form. So
yeah, if you have a base group there, it can actually help you see if it's right. And
then the other thing you have is alpha and the beta. I don't know if I should get into that right now. I have some slides I didn't know you were going to go into the carbohydrate naming, or I could have pulled up some slides on that.
The reason I brought
the
this
big
up,
picture is that we just wanted to show that there's lots of different ways that you can arrange these, and only one of them is found as part of DNA or RNA. And the paper we're going to be looking at tonight is looking at RNA, and it assumes that you get not only the hydroxyl groups pointing in the right orientation and the right number of carbons, and then they're attached to phosphate in the right way, and they have the right base attached, and then all of these things have to stay stable. Then there's the second problem of how do you get them in the right order to actually code for functional information.
Now, the reason that I point this out is, since we were talking about the math, it's easier to do math if we're analyzing at this level. It's far easier to calculate that at this level of organization. It's easier to calculate the improbabilities. Very simple. You just use the law of large numbers, the binomial distribution, things like that. It gets a little more complicated when we go to the next layer of organization, which is the subject of tonight's paper. But I was so excited to show this to you, Dr. Carter. And you know I'm a nerd if I'm like, I've been waiting 3 months to show this molecule to you and tell you what I learned.
That's great. It's a tough, carbohydrates are really tough from a chemical standpoint because there's so many varieties of them. There's just a ton of different ways that you can arrange these same atoms in various different three-dimensional orientations. And usually when I cover carbohydrates, it's like a 2-lecture long process. You know, you're looking at least 2 hours to try to explain just the basics. And you know, once you get into graduate school, they get really mean and they make you memorize the names of the different sugars. And so you look at the structure and you're like, okay, this one hydroxyl group's pointing down, and then the next one's pointing up, and then the third one's pointing down. So, okay, that's glucose. And okay, these two are pointing up, okay, or down, and then that's a different one. Maybe it's a sugar you've never even heard of before, or maybe it doesn't even exist in nature, but you could synthesize it in the lab.
And the problem is they look so similar. I mean,
that's what makes it so hard.
Yeah. So let me see if I can do this. This is the beta form. This is the alpha form. These are furanoses. I don't know what.
And how did you know it was beta and alpha? Can you explain to?
Beta means this hydroxy, the one to the attached to the 1' is what we kind of call pointed up, for lack of a better
That's
word.
Right. Mm-hmm.
So that's beta. Alpha is pointed down.
Right.
And this is really important because in a prebiotic scenario, if you start, it's very important you get the same one there, because it's just kind of like a railway. You want the railway to be uniform so the train can travel down. And in the case of DNAs and RNAs, well, DNAs especially, we want the read and write machines, the polymerases or the other machines, to be able to just traverse this railway nicely. It doesn't work so well if they're these other forms. That there has to be uniformity. And that's why I was saying it's nice that we can apply probability. So the numbers that I got, that I've gleaned from the literature, and if I'm off, I don't think I'm off by a lot. Roughly 25% of the time it's going to be in one of these two forms. These are the furanose forms. This is in water, if you have water. The others are the pyranose forms, roughly 75. This one is like less than 1%. So, I mean, I had a little bit of a rounding error there, but those are the approximate numbers. Now,
right. So what you're saying is pentagon shape versus hexagon shape, but you haven't changed the number of atoms.
Correct.
So the top, it looks like a 5-membered ring. That's because there's a 5-membered ring there. And there's 5 carbons, 1', 2', 3, 4, 5. And then the diagrams below, there's also 5 carbons. 1, 2.
Right. 1,
yeah,
2,
1, 2, 3,
5.
4, 5. Right. The way you get a 5-membered ring is you just have one of the carbons sticking up above.
Right here.
Right there. Yeah, it's
right.
not labeled.
So naturally, and this is what Change Tan was pointing out in the first edition of her book that never got published, but a lot of the concepts were in the final edition with Rob Stadler. So I read actually both manuscripts, the first edition and then the one that was written with Rob Stadler. They're both really good, but the first one is the people that would read it would be like Dr. Carter. It's not for public consumption.
Oh yeah, I breezed through it, but I was like, oh, I think some people would struggle with some of the chapters if they didn't have some background. It's so challenging because you get so used to the lingo and years and years of looking at this that you forget. It's like somebody who kind of goes back and forth between speaking Spanish and English, and you're like, oh wait, I don't know Spanish. Just keep it all English, and you're like, oh yeah, I forgot you don't know what that word means. Or
right. So this is from Dr. Tan's first manuscript, and what she was pointing out is the difficulty when they try to polymerize this. Not to mention that's also another problem to do this in water. Is you have these automatic contaminants. So Dr. Tour is talking a lot about relay synthesis. The problem is, even if you started out hypothetically, and this is actually not possible, if you started out with this form, it's going to spontaneously make all these other forms, these contaminants, especially in water. That's what they call it's an equilibrium condition when you have equilibrium concentrations of the various isomers. Is that correct?
That's right, because what's happening is every once in a while, the ring will open up and go back to that middle structure there. And when the middle structure spontaneously reacts again and forms a ring, there's a chance that it can do the 5-membered ring, the 6-membered ring, or the way that it connects, there's a chance that that hydroxyl group on carbon 1' can point up or point down. So, even when you have sugars that are made by biological organisms, like say the sugar in your ice cream, and it's sitting in your freezer for a while, that hydroxyl group in the 1' position will spontaneously go from being in the up position, the beta position, to the down position, or vice versa. And one of those is sweeter tasting than the other. So after a while, your ice cream loses its sweetness because it undergoes a type of racemization where it's spontaneously flipping from a sweeter form to a less sweet form. But you'll still have the enzymes to be able to break down that D2 deoxyribose. But that's the sugar you're going to find in your DNA. There's a lot of other sugars that are not going to be found in DNA, and if they do end up found in DNA, like in a primordial soup, where you're just, even if you could create these complex molecules, you have trouble getting the ones used by life and that are consistently the right orientation. It's like if you're trying to make a spiral staircase and they have some of the steps that are lined up just right so you can continue the spiral, but then the construction worker hands you one that was meant for a corkscrew that goes in the other direction, and you get the steps mixed up. It's like, wait a minute, first I was going around clockwise, and now it's starting to go clockwise. It's like the whole staircase is ruined. It's got to be consistently the same type of step to form like a clockwise corkscrew or a counterclockwise corkscrew. And so that's where we run into problems with having this come together when you've got a mixture of different parts, the good parts that you need, and they're going to be vastly outnumbered by the wrong shaped parts that can still participate chemically when they're trying to polymerize and form these reactions. So that's why purity is so important here, because
exactly,
you look at how similar those molecules are. Those hydroxyl groups will keep reacting, whether they're the sugar on the left or the sugar on the right, which is not what we want.
And the point I was headed for, and I'll give you the novel thing that I found that I had to search the literature. In an aqueous solution, the formation of these contaminants is inevitable. You can't. That's the thing when Dr. Tour's talking about relay synthesis, that won't even apply here, because even if you start out and it's in water, I'll tell you the killer thing I found out, because when we had that side chat about 3 months ago, I asked you, okay, so what happens when you remove the water? Does it crystallize in the same proportions? And you said it may not. And so I dug this up. It turns out without water, it's only the pyranose form.
Interesting.
Only the pyranose form. So it's like, okay, if you want to try to do dry synthesis, forget it, because your furanose form, which is your D2 beta D2 deoxyribose form, will not exist in the dry form. And I had the references in another PowerPoint set of slides, but I said I wanted to tell Dr. Carter I slogged through Google for hours to find that reference, and I'm sorry I don't
if you
have
can send that to me, that's really interesting. That the whether water is present or not will affect the outcome of what is possible. This is a good time to bring up too that one of the criticisms that or the points that abiogenesis researchers, origin of life researchers bring up, like you see more recently with Dave Farina and James Tour. Dave had some origin of life researchers on, and they talked about how, there's all this work showing how you can favor one isomer over another, because the isomer problem is also true of proteins that have to be made up of amino acids. And in, for the most part in nature, you find the left-handed amino acids favored, and you find the right-handed sugars favored. But and naturally you get, a mixture of them, a racemic mixture of, usually 50-50 left and right. Well, there's been some experiments to try to show how you could possibly favor an abundance of one stereoisomer over another. So let's say you get 60 or 70 or even 100% of left-handed amino acids or right-handed sugars. It doesn't matter, because once those become incorporated into in a polymer, like you take these sugars and link them together, or to make complex carbohydrates, or you take the nucleobases and the sugars and link them together to make nucleic acids, or you take the amino acids and you link them together to make proteins, they will spontaneously start going back into that racemic mixture. There's not a way to keep maintaining them unless you have special enzymes called racemases or epimerases that can repair them. So once they're put in that chain, they can flip-flop back and forth between the natural and the unnatural forms. And our cells have to either clean that up or fix it, because it can cause problems. It can wreak havoc in the function of a protein, or the folding of a RNA, or whether the carbohydrate can be usable or not.
Right.
Yeah, that it doesn't. Even if you could find a way to make these purified, you overcome the chirality problem, they're not going to stay in that position for very long.
Exactly. And then
things like temperature, pH, water, all those things are going to heavily influence how those start to go from pure form to the 50-50 mixture of good and bad.
And then this is the one, the reason I like this one with the other kinds of isomerizations going on, this is piled up on top of it. So not only might we be flipping between the left and the right, even though it's slower in deoxyriboses, it doesn't quite have the short half-life of amino acids. We have this other isomerization problem with the pyranosis and furanosis. And what's really bad is if you want to get a furanose, you have to put this in water. But in a water environment, you can't do, it's very hard to have a condensation reaction to polymerize it. So
right. What Sal's talking about is if you want to take, these chains and link them together, so the individual units are called monomers, and life is full of important monomers that link together to make polymers. So I already talked about some of them. We have the sugars, the simple sugars, that when they come together, they make a dimer, and then you put 3 of them together, you have a trimer. You put a couple more together, you have an oligomer. You get more than about a dozen, and we call that a polymer. Poly meaning many. So a polysaccharide or is a complex carbohydrate that's made up of a chain of sugars. And remember, the chain can also have branching points. So like starch, and which is how plants store their carbohydrates in long chains, and amylose, which is how, glycogen, which is how animals store their carbohydrates. these have long chains with a bunch of branch points. So it's like a big bushy molecule, the big complex carbohydrate. And so that's crucial for life. You know, cell membranes have these receptors made out of branch carbohydrates that make the possibilities of binding interactions much more complicated than if you just had a simple membrane or even protein sticking out of that membrane. A lot of the lipids and a lot of the proteins in membranes have these complex carbohydrates attached to them that make the surface even more fancy and more sticky and more complex for all the different three-dimensional interactions that it can have with another cell. And we're still trying to learn about that whole sugar code and how these structures form complex functions. And then another kind of polymer we have is the nucleic acids. And so that's what we're going to talk about in this paper, taking the 4 bases, whether it's RNA or DNA bases, and linking them together to make long chains of RNA or DNA. And so RNA and DNA, these nucleic acids, are polymers. And then another kind of polymer we have is where we link together the amino acids to make polypeptides, and then they fold into proteins. So in all cases, when you're taking those individual monomers and linking them together, in order to do that, it's a condensation reaction. Water actually comes out of there. So that's what Sal was talking about. Whenever you're linking these together, water is ejected from the molecule. But if it's happening in an aqueous environment, the tendency is to not make more water when it's already surrounded by water. The tendency is to do the reverse, where some of those water molecules are going to take that chain and split it, which we call hydrolysis, using water to split long chains apart into their individual bases. And this happens naturally. Just, a dead body in a lake or river or ocean is going to decompose a lot faster than in the desert, because the water helps speed that up. When you steam or boil your vegetables, and the starches, they loosen up because the water is hydrolyzing it. And you have way more water in these environments than you do proteins or carbohydrates, and so the water is always going to have the tendency to break it up unless you can really dry it out. But once you dry it out, guess what? Then your chemical reactions that need water to take place stop as well. So it's like, even if you could remove the water temporarily, in a wet dry cycle, or get it to polymerize on a clay, eventually you got to get those molecules back off the clay or back off of dry land and back in the water so they can do the kind of chemical reactions that we need water for. I mean, water is essential for life. At the same time, it also has a tendency to destroy the molecules of life. So the only way we can overcome that is to speed up the building faster than the breakdown by using enzymes that can speed up those reactions by, sometimes thousands or millions of fold faster than they would happen naturally without the enzymes.
Right. And then in many cases, you would add energy with ATP.
Excellent point. Yeah. Just because you put these molecules and mix them together doesn't mean they're always going to link up. It's actually not that favorable. You're going to have to require not only energy, but the intelligent use of energy. It's got to be, these molecules have to be held in place long enough and oriented in the right position so that their covalent bonds weaken and then break and then reattach to other atoms. And so that's what the job of the enzyme is to create this three-dimensional structure, this pocket, to hold that molecule, the molecules next to each other long enough and in the right angle and orientation, plus sometimes some energy like ATP to rearrange the electrons and form new bonds or to link them up.
Because in that aqueous environment, you have to have a favorable Gibbs energy for the reaction to go forward to get that condensation reaction. Otherwise, you're not going to get a reaction at all. But the point I was making here with this diagram is, if we have these wet dry cycles, in the dry cycle, the very molecule you want disappears. It becomes this. The furanose will become a pyranose. And that's what I want. I waited 3 months to share that with you. I said, this is just another barrier. And then I looked at all the ways that they do DNA synthesis. And like Dr. Tur said, is they have to, it's a little more stable if you have the ribose with the phospho group. or something, or a full a nucleotide with all the protection groups or something. You can't just do this. It's very hard to make this into a nucleotide. You can make it into a nucleotide, but you won't get this. You won't have these stable. You won't have the beta D2 deoxyribose as your sugar.
Right. If everyone looks at the structures on the bottom, you see how there's 3 OHs there. That means the next sugar and phosphate that come along has a choice to bind between any 3 of those, but only one of them is the right choice. So you're reducing your odds of getting these things to line right up and link up every single step of the way. So if you've got 100 base pair long RNA, that means it's got to get the right hydroxyl group to link the phosphate to every single time. And with DNA, it's even, or with RNA, it's even worse because there's another hydroxyl group on that number 2 carbon. So there's 4 choices there that it could bind to. So that's why when they do it in the lab, they've got to block those sites temporarily by putting another chemical on there so that only the right hydroxyl group is reacted, and then go back later and then remove those other blocking groups so that it looks like an actual DNA or RNA molecule.
Right. And I found the method that they're able to identify. They have to use PAGE or HPLC because so they could actually figure out which one had the correct protecting group. I mean, this is, the more I saw this, I said, this will not happen in a random environment like a prebiotic soup. And when you're speaking about water, and you said it'll destroy the reaction, but you also need it to make the reaction, they call that the water paradox.
Right.
So this is known in, that was mentioned in Change Tan's book. But James Tour, he always has a little bit of sharp humor because he's talking about the problem of water. He said, what are you going to do? Are you going to remove the ocean? So all that said, we haven't even gotten to the paper. The paper assumes that all these problems have been taken care of somehow.
Which is a really important point because there's a lot of assumptions made in this paper. And you're going to see that even if everything goes smooth, the odds of getting the right bases in the right sequence to do something functional is still so low that it's beyond the probability for all of the atoms and all the time in our visible universe. So by the end of the paper, they have to invoke some hypothetical situations, which of course takes us outside of the realm of science, which means that no matter what your worldview is, there's going to be some leaps of faith here, whether you think God has created life or you think that somehow nature finds a way. It's going to require going outside of the realm of science because we're going to be, the conclusion of the paper is that we have to leave the observable empirical realm and go into the unobservable or non-observed possibilities and invoke things like multiverses in order to overcome the low probabilities that are acknowledged in this paper.
Well, I thank you for bearing with me. I know we were going to talk about the paper, but I was waiting 3 months to show you about those pyranoses. And
I'm so glad you did. Yeah. So if you haven't been able to follow along so far, it's okay because we're going to go into a whole nother related topic. But now you can stretch, take a breath, get ready for the part 2 of our talk where we're going to go through the paper. I don't know if we need to go through every single word of the paper or just cover some of the highlights.
Well, thankfully it's a short paper because if it's like 2 pages, the one that Emery and I tried to tackle was like 21, and that was 5.5 nights to get. Yeah,
I'm still on like night 3 or 4 or something. I'm still trying to catch up even at 2x speed.
Yeah. And I do want to thank everyone who was forbearing with that. And I wanted to say this stuff is
great
not easy.
paper.
God is not just to relate this to the Christian faith. I was talking about the hidden God, and one of the classic examples of that is in Luke. I think it's chapter 24, where it talks about your car, someone's car.
The neighbor's car alarm. Let me mute.
Oh, so the road to Emmaus, where these two disciples, one of them named Cleopas, was talking to Jesus, but they were not enabled to see that it was the Lord. And so sometimes, God will conceal, but then he'll talk to you still, he'll communicate, he'll teach you. And then eventually you see. And so how this relates to what we're struggling through and, slugging through, sometimes we're going to have to do a lot of digging to be able to find some of the evidences of God. And it could be a long time. And that's why the Lord says, seek him with all your heart, because, when I nearly left the Christian faith, it was a study of abiogenesis that really started to spark my return. The first spark was when I asked the atheist, I said, okay, what do you know? What is your basis of meaning? How do you value things as good and bad? And they couldn't give an answer. And I said, I'll take my chances then. I'm going to go back to the Christian faith, because you don't have any evidence and you don't have any answers. And that was what Randolph Richardson recently said. There are no answers in atheism. That's a slight paraphrase of his original wording, but that's close enough. And I did a show on Richardson's principle. So the next step was studying abiogenesis. So I really appreciate everyone that has been studying this topic. And I could tell that when James Tour was giving his 15-part series, everyone was just praising the Lord. And that's just beautiful to see people studying chemistry and praising the Lord after. Now, the funny thing that Emery Moyna said, he said, Sal, you were studying biochemistry and you're sitting in class praising the Lord. He said, when I was studying biochemistry, I was just praying that I could get through the class. So, okay, that was a moment of humor. Now, the title of the talk, if I could just talk a little bit about this inflationary universe. This is one reason when I was making my journey from old earth creationism, I was halfway convinced of a young cosmos, and I became a young cosmos creationist when I studied inflationary theory in graduate school at Johns Hopkins. I was studying general relativity, cosmology, astrophysics. And I almost fell out of my chair when they're describing the mechanics of this. And I've given 2 shows where the scientists have said, yeah, this is falling apart. The Big Bang, right now, this is one of the ways that they can fix it. The other alternative is variable speed of light, which of course I'm friendly to. So that's a little bit. So this inflationary universe is on shaky ground. And Dr. Carter, if I may ask, I know you're a SDA is your theological background. I've actually heard that there's some traditions, and I'm really not familiar with this, that believe the universe is old, but the earth is young, or the false, like Sean Pittman might be a young life creationist, and can you explain that, what the viewpoints are? And I'm not trying to put words in your mouth. I actually don't know. What the
I know Sean.
are.
He's a buddy of mine. In fact, I want to get him to come on some of these shows sometime. But with some of the Adventism, the question of the age of the universe isn't a part of our doctrine. So it's either way you can believe old universe, young universe. But what absolutely is crucial to our faith is young life on Earth. So like young biosphere creationist, maybe even young solar system. Because the Sabbath being, the reason why it's remembered as part of the 4th commandment written by the finger of God it's a call back to creation week. Because if you look at the commandments, it says 4 and 6 days God created the heavens, earth, sea, and all that in them is, and so it's absolutely tied into the Sabbath, and it's crucial to our faith that. As Seventh-day Adventists, that we believe that life has a recent origin. I'm still kind of unclear myself how I feel, because I've heard such good arguments on both sides for an older universe. Like when did the whole battle in heaven where a third of the angels were cast out, did that happen before Adam and Eve, or when did that take place? It's hard to imagine that an all-powerful, all-knowing being would just twiddle his thumbs for eons and then only create life, 6,000 years ago on one planet. I tend to think that there's many other created worlds that they're way beyond the reach of our polluted, sin-tainted, rebellious planet, and that God interacts and loves those creation too. But that's not something I can point to a verse and say. It's just because that's how I think an all-powerful God would be, is that he has all sorts of he made a very large universe, and it just seems strange that he would only have life on this one planet. But again, I could be totally convinced by the studies I've seen that the entire universe actually is recent, and the time being relative, that it's not like we have this impatient God that sat there in complete darkness and emptiness for trillions of years or something, that and the time is relative. So yeah, it doesn't concern me as much, the age of the universe, because it doesn't bother me as much theologically as it does if God used evolution to create life. That would really bother me, because I believe God is omnipotent, and if God is all powerful, in other words, he was perfectly capable of creating something as complex as humans in a day, but chose to use this long, drawn out, circuitous method that requires that's super sloppy and somewhat unpredictable in terms of like which organisms are going to survive and which ones are going to go extinct, and only to at some point say, actually, death is wrong and death is the wages of sin, and even though it's the useful tool for creating life, but all of a sudden now it's bad. And so that kind of a God that's all powerful and used evolution to create life, even if he had to intervene at several different points, like, okay, yeah, we can't get the first cell, so I'll create that, or we couldn't get eukaryotes from prokaryotic precursors, so he helped there. And then and then finally comes down to like ape-like ancestor, and this, finally there's something intelligent enough to worship me as creator and give me credit as creator, even though I really didn't have that much to do with it except intervening a few points here and there. To me, that would be the most frightening thing imaginable, is an all-powerful God who was cruel and lazy and didn't just create us right away. And the other option is, what if he wasn't powerful enough to create humans in an instant? Then I would worry about resurrection, including the resurrection of Jesus. If he couldn't create humans in a day 6,000 years ago, how is he going to recreate life on resurrection day? Or how do we know that he even resurrected Jesus? It's not an easy feat, even if you have all the parts to create life.
Well, again, part of the reason I asked that was I really genuinely wanted to know, because from the like Loam and Linda, et cetera, they haven't really talked much about the universe. But since that was my field, cosmology, I was just curious, and I said, I think that there are some, like, I think I talked to Paul Guillem, and he said his faith tradition said that he's not committed to
that's right,
the universe being young too. And that's the first time I heard that. Now, the reason that's of interest to me, I often say, okay, I'll tell you what my beliefs are, but we can make models like we assume certain things just for the sake of argument. And one of the models that I like is that the universe is, let's just assume the universe is old. We can make arguments that human life is young or other life forms are young. And so that was the main thing. But how this ties into the paper is that it does talk about the inflationary cosmology. And I just needed to point out, on scientific grounds alone, we don't have to talk about theology. There is some problem with the inflationary universe, and we probably will see more. But while you're here, it's like, yeah, this is something I've been dying to also ask Dr. Carter what the spectrum of viewpoints are on the age of the universe, because like the evangelical young Earth creationists, most of them are also young cosmos creationists. I don't know I don't know personally of any. They may have introduced themselves to me, but I don't know of any evangelical, which is my theological background, that will be a young Earth creationist and also an old Earth universe, an old universe. They're either, they're also almost, that I can remember, they're all young universe creationists. And I'm like, well, then in that case, I would call you a young cosmos creationist. The reason I don't say young universe creationist, that's the acronym would be YUC, which is yuck. And I'm like, no, I'm not going to use that acronym.
I've heard people say young biosphere creationists. So the short answer is we're basically agnostic to the age of the universe, and I guess probably why we haven't spent so much time trying to figure it out either theologically or scientifically is because it doesn't seem to affect the outcome in terms of like salvational issues or doctrinal issues as much as young life or young biosphere creation does.
Yes. So that's why what I focus on a lot on the channel is I would call it, the term is young biosphere creationists.
Right.
So a young cosmos creationist would by default also be a young biosphere creationist, but we can argue young, and so you would classify yourself as a young biosphere creationist, and so would Shaw Pittman. Is that fair?
Sure. And again, we're up for changing our minds when, it's just at this point I've seen good arguments on both sides, and so I just haven't making it a decision, and not sure that I need to commit to a decision unless someone can show me a reason why it would be offensive to God or something to believe either way. And again, I just haven't studied enough of the cosmology and physics, and I'm fascinated by it. I just haven't spent enough time studying. I'm more concerned with trying to get the idea that God created something as complex as humans. He's powerful enough to create a human being in an instant or near instant.
Well, I could tell you this. I can imagine you're busy enough doing the chemistry side. So, and I certainly, and again, one reason I tend to like to make young biospheric arguments, and I try to caution my evangelical young cosmos, my evangelical young Earth creationists who also believe the cosmos is young. We have really good arguments against abiogenesis at the present time. And I can't say that we have anywhere near that when we're dealing with cosmology. There are too many unknowns. Right. And so, I'll tell people what I believe, but I'm just like, well, it's not like when I can argue abiogenesis and show these like Pyranose diagrams and stuff and say, don't expect this to happen. I can't, if I had a conversation, quasi debate with an astrophysicist or cosmologist, I would really struggle. It wouldn't be the same with an origin of life researcher. It'd be totally, best I'd be praying we could have a draw because, I don't have as many facts available. Now, I will say the trend has been to a little bit more anarchy as far as the Big Bang, because even the secular world is beginning to question it. So there's been cracks. There have been professors of mine that were skeptical of the Big Bang, or if there weren't professors, they were professors at the university I attended. So now
that's what's fascinating to me is that, something that's like you think is so set in stone, and it's basically like a law or just a principle of physics that there was a Big Bang, some kind of expansion of it, and that even the non-theistic cosmologists are starting to question some of the models. That must be how people feel about, when we look at abiogenesis research and they think, oh, isn't that all figured out? It's like, no. There's so many things that we still have to try to figure out. And there's more than just a few cracks in the theory of abiogenesis. So it's fascinating to me when you find out that there's a field that's outside of your own and you think that it's all solved, and that the experts are starting to get new evidence that makes them question it. That makes me really interested to learn more. But I just wanted to comment what George said about death before sin. I totally agree. I do not think that there was death as a mechanism for filtering out the weak so that the strong can survive. That's my biggest problem with having like a theistic evolution approach where there was, years and years of death and suffering before sin. I think the wages of sin is death, and that prior to the fall of Adam and Eve, and that there wasn't a animal or human death.
Okay, I think we can start with the paper now, and then if there are open issues, we can address them later. But I it's been an hour before we haven't even started the paper. But this
I told my wife I probably won't be more than an hour, and I'm like, oops, we're just now getting started.
Oh wow, I'm sorry about that. Well, I can tell you, if we have to resume this at another time. But you have given me first off, tremendous gratitude, because I have a debate coming up with James Downard on Modern Day Debate, and people know my feelings about it. I normally don't like to debate, but James Coons, who runs the channel, has been a supporter of mine, and as a favor to him, I'm gonna do a debate, and plus I show I'm not afraid to engage the other side. That's important. That I don't want to be accused that I'm not going to face my critics. Now, I can't face all of them. On Reddit, I've blocked 100 of them. I'm just like, I'm not going to have time for 100 of you guys. And you're all going to ask me the same question. I'll give an answer to one of you. You'll ignore it, and you'll just ask me the same question again. And then I have multiply that by 100. And just like, I'm sorry, guys. So, this discussion this last hour has helped solidify my confidence for that. So now, I'm happy to just kind of read this paragraph by paragraph, because it's a short paper.
Yeah, or even at least the sections that I highlighted there. I wanted to just give a little bit of, go ahead and read this first part of the abstract so that we can give people an idea of what it's about, and then I'll take a moment to explain why the RNA needs to be a certain length in order to have function.
Okay, so this is a relatively recent paper. This is this year, 2021, I believe. So the title of the paper is Emergence of Life in an Inflationary Universe. And that is Tomonori Totani. So I'm presuming that sounds Japanese. Abiotic emergence of ordered information stored in the form of RNA is an important unresolved problem concerning the origin of life. A polymer longer than 40 to 100 nucleotides is necessary to expect a self-replicating activity. But the formation of such a long polymer having a correct nucleotide sequence by random reactions seems statistically unlikely. However, our universe, created by a single inflation event, likely includes more than 10 to the 100 sun-like stars. If life can emerge at least once in such a large volume, it is not a contradiction with our observation of life on Earth. Even if the expected number of abiogenesis events is negligibly small within the observable universe that contains only 10 to the 22nd stars. Here, a quantitative relation is derived between the minimum RNA length L-min required to be the first biological polymer and the universe size necessary to expect the formation of such a long and active RNA by randomly adding monomers. It is then shown that an active RNA can indeed be produced somewhere in an inflationary universe, giving a solution to the abiotic polymerization problem. On the other hand, L-min must be shorter than 20 nucleotides for the abiogenesis probability close to unity on a terrestrial planet. But a self-replicating activity is not expected for such a short RNA. Therefore, if extraterrestrial organisms of a different origin from those on Earth are discovered in the future, it would imply an unknown mechanism at work to polymerize nucleotides much faster than random statistical processes.
Okay, so we can summarize that in non-technical jargon. Before we knew that RNA could do more than just carry some information, that actually some RNA could fold in such a way like proteins fold and have an enzyme-like function. We call those ribozymes. Before that, we just knew that proteins were the things that when you link them together in the right order of amino acids, that they could fold, not always spontaneously. Sometimes it takes the help of assistance from other proteins to help fold them in the right shape, like chaperones. But when they could form it in a three-dimensional shape, they would be able to perform a chemical reaction, or speed up a reaction, make a reaction happen that wouldn't normally happen, or would happen so slow that it wouldn't be helpful to life. And then a couple decades ago, it was discovered that there's actually RNA that can have similar function. And these RNA strands can fold in such a way that they will have an enzyme-like function, and they're called ribozymes. So that got the origin of life researchers all excited. They thought, hey, maybe we don't have to have the proteins first. We can have RNA, which will serve not only as a template for storing information, like a genome, an RNA-based genome, and there are some organisms like RNA viruses that store their genetic information on RNA rather than DNA. And this RNA can fold in such a way that it speeds up chemical reactions needed for life. The problem is you have to have a chain of RNA that's long enough that it actually can form into a three-dimensional structure. So I borrowed some of my kids' toys here. These are called bunchums, and I stuck them on a pipe cleaner here. And if you let all the pink ones be, say, like adenine, and adenine binds to thymine, or in this case, it'd be uracil since it's RNA. So we'll say that the pink and purple will bind to each other, and the yellow and green will bind to each other. Adenine and uracil will bind to each other with 2 hydrogen bonds, which are kind of weak. So these things are kind of sticky, like hydrogen bonds would be. They're not that strong. And then the guanine and cytosine can stick to each other with 3 hydrogen bonds. So you have to have a right sequence in order to get these things to fold in water spontaneously, so that it'll form things like loops. Now, if you see, I can bring these together because the green and the yellow cooperate with each other. They happen to have the right three-dimensional shape to where they can form some hydrogen bonds where they all line up, either 2 or 3 of them. So the yellow and green line up, and the pink and purple line up, and it just so happens that they're in the right order, that when I form this hairpin loop, they will stick to each other at a low enough temperature. If it gets close to above 95 degrees Celsius, and you're close to boiling temperature, remember, above absolute zero, all these atoms are moving, these molecules are vibrating, and there's too much energy and they'll come apart. But once you bring the temperature a little cooler, they'll slow down enough so that they will stick to each other. And this part won't, because it's not, there's not opposite interactions. They're not the right orientation. There's not the right positioning for these to stick to each other. So these won't stick to each other, but where you have the opposites attracting, the A and the U and the G and the C, you'll get a nice shape here. So let me share the screen here to show you kind of what it would look like, why you need a certain size. Okay, let's do this one, share. Okay, so this is from a paper in Nature Chemical Biology, the crystal structure of the Varkud satellite ribozyme. It's got a nice diagram here showing how you can get 2 different RNAs to interact with each other and fold. And where you see these little dashed lines is where they will fold. They will hydrogen bond to each other, and these hairpin loops are where they don't bind to each other. And it folds in 3-dimensional space and looks something like
Dr. Carter,
this.
I'm sorry. Can you zoom in just a little bit? I'm sorry to interrupt you.
No problem.
It's just so that the viewers and I can see it a little clearer. Thank you.
Okay, let's see. Let's get this up a little bit more. I think that's as much as I can zoom in. Yeah, so it looks kind of like a tangled web here, but everywhere you see these little sticks coming off the main branch, this backbone here is going to be your sugar phosphate backbone, and then the bases are going to be these little sticks facing away from it, and they are going to hydrogen bond with another matching pair on another strand, and it will fold into this 3-dimensional shape that will have some kind of function. Maybe it'll break apart another molecule, maybe it'll take 2 chemicals and stick them together and, do something. There's a limitation to how much they can do. Obviously, proteins are better at this job because they're stronger, they're more stable, they're less flimsy. And so for the most part, it's proteins that are going to do this job. But there is a way for RNA to do this job, but it's going to take a certain length to accomplish this. Typically, like the very shortest ribozymes might be like 100 nucleotides long. So, it's going to, if you imagine like all of these bases here, you couldn't get by with a ribozyme that was just this short. They just had like, a dozen or so nucleobases. You need, or nucleotides, sorry. You need a lot more than that in order to get a fancy three-dimensional molecule that has some kind of function that will do things like make copies of molecules or link molecules together, break molecules apart. And so this paper is pointing out that, to get a simple ribozyme is going to require, they estimate a minimum of 40 nucleotides linked together in the right order so that it folds into a three-dimensional shape that will perform a sped up chemical reaction. And 40 is being a very on the low side. Realistically, it's probably more like 100. But we'll see what the chances are of even getting, say, 20 or 30 bases, nucleotides to link up in the right order to perform a function. Because there's only a limited number of ways that you can arrange these to have them function. For the most part, just randomly putting together chains of these nucleotides, there's a lot more things that will fold and do nothing compared to the number of ways that they will fold and perform a chemical, an enzymatic-like function.
Just briefly, when we were on Otangela's channel with a gentleman named Dak or Donald A. Kronos, Molecular Alchemist was in the side chat. He said, there's a ribozyme made of 5 nucleotides, and you had a good answer to that.
Part of it is it's only beneficial if it's interacting with something that's much bigger. Being 5 nucleotides by itself is not going to be very helpful. Unless it's interacting or binding to something else that is a lot more complicated. So if you look at the substrate that enzymes bind to, they can be really small. It can be just a carbon dioxide molecule, but the whole rest of the molecule can be, hundreds or thousands of base pairs long just to be able to get the right three-dimensional shape to hold on to that molecule or link 2 of them together. And so you're not going to have one short molecule interacting with another short molecule and doing very much. You can have proteins that are only like 9 amino acids long. They're called nonapeptides, like certain hormones, oxytocin, for instance. And that's very short. You're like, hey, that's only 9 amino acids. That's possible, isn't it? Well, think about what it binds to. It binds to an oxytocin receptor, which is very complex, may even have some sugar groups attached to it to make it even more complicated in its three-dimensional structure. And then it's not just a matter of binding to it. It's got to have some kind of effect through the membrane where it causes a series of chain reactions inside of the cell, like a phosphorylation event or something. And then that's got to make a signal go all the way to the nucleus or to the DNA and turn on and turn off certain genes. And so there's a whole array of things that have to be much more complicated and work together in harmony. It's not just a matter of having a tiny little molecule do one little thing and calling that life.
I know that might have been trivial for you, but he actually did stump me. Molecular Alchemist stumped me with that. Like, I need to ask Dr. Carter. And so Thanks for reminding me. The other thing is, just because it can catalyze a reaction and it's only 5 nucleotides doesn't mean it's going to be able, that the system could be, the 5 nucleotides could be evolved to do something else that's useful. There's no guarantee.
That's right.
Yeah.
In fact, you're probably more likely to accidentally fold something that is destructive. And there are ribozymes that chop up other RNAs or chop up DNA or do other things like that that could be catastrophic. If the first ribozyme on the scene in a primordial soup or a pond or a geothermal vent or whatever just happens to fold in a way that chops things to pieces, that's would
we call it like an endonuclease?
That's right. If it chops in the middle of nucleic acid, it's called an endonuclease. If it removes things off the end one at a time, that's an exonuclease off of the end. So yeah, whether it's an exonuclease or an endonuclease, you've got something that suddenly speeds up the destruction of neighboring molecules that are waiting to do something useful for life by thousands of fold faster than they will form. So maybe even millions of fold time faster than they can self-polymerize. So it's like opening up Pandora's box if you accidentally get a cleaving. And just through experience, it's usually easier to create things that destroy than create. It's a lot. You're more likely to get a knife than epoxy.
Let me see if I can.
The other thing I want to point out. Go ahead and finish your thought, because I'm going to transition a little bit here.
To that end, I made a video that was criticizing Professor Dave, and I have I pointed out what you said, but that wasn't original to me. That was Rob Stadler and Change Tan. They said that we could end up creating these cleavers. I called them cleavers.
That's right.
I made a video cartoon, and people actually complimented on me. Since you're working with animators, you kind of know how hard it is to make a cartoon. It took us 6 weeks. No, it took us months. I mean, on and off, not full time. And we had our animation team and the actors. But anyway, we had that one scene, and I'm providing it in the link. Give me a moment to type it in. Cartoon by Sal on Professor Dave. And it's only like 5 minutes long. And what we did, and you can see it, is we first said, okay, so the first RNAs will be cleavers, and we showed pictures of cleavers. So these are your nucleases. And then I said, if that's going to be the case, it's going to be like the Texas Chainsaw Massacre. We showed some guy with his chainsaw just chopping. Okay, so
that chainsaw has an almost unending supply of gasoline. It's only until the chainsaw breaks that it's going to stop working, or something goes wrong with it. So it's going to keep chopping over and over for hours or even days until it starts to decompose spontaneously. And think of all the damage it can do. Years worth of assembling all these beautiful molecules that haven't come together and done anything useful yet are going to be destroyed in that process. The other thing I want to point out is the whole reason we had that whole hour-long spiel in the beginning is because what I'm showing here are only the nucleotides that are useful for life. But what if there was a whole bunch of other molecules that could also fit on here, these black ones here, that have nothing to do with life, or they actually interfere with the next reaction? Like maybe this black molecule here prevents further adding of nucleotides. So that's the end of the chain there. It can't grow anymore. Or this molecule will not interact with any one of these other ones and help it form. And and what we know both empirically and through in silico experiments, and what we find in meteorites, is the number of these useless or detrimental black molecules here outnumber the ones you find in life by at least 1,000-fold, maybe hundreds of thousands or millions or billions. Even Cronin said that, there could be 100 trillion molecules that have nothing to do with life. We call them abiotic molecules for every one of the biotic, the ones that would be useful to life. So imagine trying to randomly put together this sequence, and I'm just blindly, putting some of these chemicals on the end here and hoping to get them in the right order in a way that it's going to fold. But in my pile, I've got 1,000 times as many of these black pieces as I do the colored pieces. What are the chances that I'm going to get a nice long straight chain of 100 of these together that are all the pinks and blues and greens and yellows without a single black one ending up there, even though in the pile there's 1,000 times as many black ones as the colored ones.
And it may not
and
even
all it takes is one of these black ones to mess it up.
Yeah, and it may not even have that linear polymer shape. You could end up growing all sorts of funny looking chains.
And
Goodness,
if you want to make copies of this, you need it to be a nice linear chain. But what happens in a dirty primordial soup is you've got, you're just this huge abundance of other molecules that will attach itself and ruin that molecule. So now instead of having a nice thing that will fold, it'd be like, oh no, I'm stuck in basically tar, and I'll never be able to do anything until it falls off again. If it ever does. Sometimes those reactions are so strong, they'll be covalent that they're gonna ruin this molecule. It's like we call it the chemical toxins. It ruins the function of the protein. And good luck getting it to replicate if it's coated with, like it's been rolling around in tumbleweeds or something. And how are you going to get a nice enzyme to come along and feed this through the opening of an enzyme and make copies of itself? And it's like, oh, great, I got caught up on whatever this chemical junk is that has nicely attached itself irreverently to the molecule that I was hoping to copy. So I guess we have to throw that one away and start over.
And when you talked about it becoming tar, Steve Benner's term, which was highlighted in Change 10 in Rob Stadler's book, was the asphalt paradox. It's not literally an asphalt, but that's where it heads. And I threw this on it just in passing. I pointed that out in a debate, and the guy was T-Jump, and he has no chemistry background. And he just said, no. It just, over millions of years, it's just going to build it. That's what the origin of life researchers' papers are. And it's like, oh, man.
That's the thing. The time makes it worse, because the more time you have, the more chances you have to gunk up these beautiful molecules, and the more chances you have, like, even if I could get this right sequence, like maybe even 20 of these nucleotides long, what's going to happen way before that is water and hundreds of other chemicals are just going to cut this up into pieces or chemically modify it or ruin it. It's not like I can just make this beautiful molecule and save it in a almost absolute or liquid nitrogen temperatures or something and just protect it in this little safe bubble until it's ready for life. It's going to be prone to further chemical degradation. Ultraviolet light will destroy it in no time. I mean, especially the nucleic acids. I mean, there's a reason why the RNA vaccines are stored at minus 80 degrees Celsius, because the RNA is so fragile.
Yeah.
Even this part is fragile. The nucleotide with the phosphate on it. I mean, we're talking about a matter of like hours or days or maybe a few weeks at the most that this will stay in its proper chemical structure in an aqueous solution. And again, if you dry it out, how is it going to participate in the reaction you need to find its other partners and link up into a chain?
So we have the asphalt paradox and the water paradox.
And there's a dilution problem, and there's compartmentalization and sequestration. So let's go ahead and
that's why Stairway to Life is just so great. It'll just keep hammering that, and at least I was able to add my little tiny contribution. I wanted, maybe I'll go tell Change 10 and Rob Stadler, look what I found out about those pyranoses in the dry state. You don't even get the beta furanose that you need. So that's adding insult to injury. So actually, to be perfectly honest, I would have to read this again to kind of understand what he was trying to say. I mean, I kind of see like, if we're okay, can we go back to the paper or?
Oh yeah, please. Yeah.
Okay. He says our universe created by a single inflation event. And I pointed out that this is still a Nobel Prize winner, Roger Penrose, has now questioned it. So a lot of people are even questioning this assumption. But let's just grant it for the sake of argument that a single inflation event happened. Likely includes more than 10 to the 100 sun-like stars. In fact, actually no one knows what the figure could be. They'll just say infinite. And of course the joke is, all right, we could also be in, if you're going to admit inflation and you just admit any probability you want, it's perfectly probable then that Jesus walked on water, perfectly probable in some universe that the creationists are right. So this, I just need to point out, if they're going to admit astronomical astronomical multiverses where anything practically is possible, you can start to admit other kinds of miracles too. So
you could end up with a universe where an all-powerful being exists.
Yeah.
How do we?
Yeah.
That could just be ours. Hey, there's a God.
Yeah. So I'm just pointing out that but we can make fun of the paper. But the reason I presume that this was interest to you and also James Tour, he cited it in his series. This specific paper is that it at least attempts to put figures to the probability scores. And this is a philosophical question, and I put it in my debates. I said, how improbable does something have to be before you accept the possibility that it's intelligent design or some intelligence or a creator God did it? And some will say there's nothing that would persuade me. And at least they're being straight up with that. So the probability arguments don't matter to them anyway. But there is a large number of us. It's like, okay, there's a point that it's just it's harder to be an atheist than it is to be a theist, and I fall into that category. When I see odds like that, I said that's not anything I'm going to wager my soul on. And I framed the question in other ways, like saying, okay, if you were that blind man in John chapter 9 and Jesus healed you, would you follow him? And a lot of people said, yeah, even if that happened to me, I wouldn't follow it. I said, okay, at least we know where you're all coming from. So the value of this paper for me is that it's at least trying to put some figures.
Yeah. And if someone comes away from this like, okay, I guess my faith is incredibly strong or based on something that's not very foundational in science and probability, but I'm still, so it looks like it's more likely that an intelligent being exists, but I'm still not going to worship him. And that's more of a theological problem that they have. They have some serious misunderstandings of the character nature of God. And that's a whole nother story that I think we also need to help meet the, buttress against the other end. We can talk about the probability and the chemical and physical problems with having life originate without intelligence. And on the other end, we have to be able to say, okay, what is it? Is it just an argument from statistics here? We also want to be inviting on the other end and say, okay, if that God exists, what is that God like? And when people already have this idea that he's just this cruel, absent person that reminds them of their abusive father or something, that puts them in an extremely uncomfortable position. It's not just like they have to give up believing something and admitting that they were wrong about what they thought. It's not a very cozy position for them to switch their worldview, because they're jumping into something that seems just as awful. This idea that God exists, but he's not an awesome God, or he's not a loving God, or he's an unfair God or unjust God, or they look at this world now and they think, well, how can a loving God allow this and this and this happen, not realizing that this world we exist in now is not the world that God intended, and he will do away with the way things are going now and recreate a much better world where things are operating the way they were intended to. So don't take note of how things are on earth now and think that that's what God desires or intends, or how it's going to stay forever. Because a lot of these arguments I hear, they're looking at what we see today and thinking that that's all how God intended it, and that's how it's always going to be. Or they think we're just going to be up in heaven playing harps on a cloud for trillions of years, bored out of our mind. And it's like, there's so much more we could talk about that. But I just want to bring it up now because I realized after going through this, some people might realize, okay, yeah, the chances of life originating from a super so low that we're having to invoke unobserved a universe, like a portion of the universe that is much greater than what we now see. 10 to the 22 stars in the observable universe, there's a reason why he's invoking 10 to 100 stars, because he realizes even with all the probability, all the possible chemical interactions that can happen 10 to the 22 stars isn't enough. I mean, do you even understand what 10 to the 22 stars is? It's like, go to the beach sometime and pick up a handful of sand and imagine every one of those as a star, that's 1,000,000 times bigger than our Earth or more, and has some planets or not. And then think of all the sand on all the beaches of the world, and that's what we're talking about with 10 to the 22. and
Exactly.
that's still not enough to get a short little RNA to be in the correct nucleotide sequence to form a functional ribozyme. So when it hits you that this just cannot happen by any mathematical means unless you invoke way more of the universe that we can't observe and probably will never be able to observe, or multiverses as your solution, you're out of the realm of science at that point, and you're putting your hope on such low odds that
Exactly. Right.
You got to make a choice. Do you want to live your life? Do you want to make your choices? Do you want to put your hope and faith in something that has that low of a probability, or do you want to look at some other options? And I hope that as the Christian community, we can make those other options a lot more enticing than unfortunately we have been. Sometimes it's like people say, hey, I don't want to spend eternity with jerks like you in heaven. It's like, oh, I'm sorry. Like, you have a great misunderstanding of what heaven is going to be like. And I can understand why you wouldn't want to love a God that is like the God that you're picturing or misinterpreting from scripture, or because you had some bad interactions with some Christians at church or something that you're going to blame God now on this, or you think that that's what heaven's going to be like. Let's put those rumors to rest.
And the reason I highlighted, and I'm going to echo something you just said, and actually you anticipated what I was going to say. We looked at the 10 to the 22 stars. That is what we can detect with our telescopes, so forth. And beyond that, as a matter of principle, we can't. If we assume the Big Bang cosmology, even if we built better telescopes, we're not going to see anything beyond those 10 to the 22 stars, because I think it's called the horizon problem. And I'm
Right.
embarrassed because I should know this, but that wasn't what I focused on in cosmology. We were solving all these math integrals, and I'm just like, how's this? You talk about really being deep in the weeds. And my professor, we all kind of laugh. He said he's a closet mathematician because all we do is math. We don't actually study physics. I mean, we do the math of the physics, but sometimes it got decoupled from like, what are we really doing this for? Like, I remember even with Mathematica, Wolfram Mathematica, he gave us an integral to solve. I'm just like, okay, I know this is important to astrophysics, but can't you just like say, okay, let the mathematician solve it. We'll just accept it. He said, no, I want you to prove the integral for this equation is this. Even with the computer and the computer doing parts of the integrals, it took me a whole day. So that's my excuse for not quite understanding the statement of the horizon problem. But what I was going back to this, if I understood the horizon problem correctly, even 10 to the 22nd is all that we have a limit. We are assuming that there's going to be 10 to the 100th and actually more, because no one really knows the figure. It could be infinite. And some people like infinity because then you get even more chances. Because when if the author realized, we're going to need kind of even more, rolls of the dice, the casino needs to grant us more rolls of the dice before we go broke, because life is going to be way more complicated than this 100 nucleotide self-replicating enzyme, ribozyme. But what you pointed out, and I thoroughly agree, this is now beyond science, because you cannot observe it, you cannot verify it, you cannot test it. You're just speculating. And I pointed out also earlier that the whole inflation model, that's now in doubt. So this is on extremely thin ice on many grounds scientifically. Not even the theological things aside, it's on thin ice, except for the fact he's going through and calculating some probabilities for us within our current, what we can estimate within our own universe. So it has value there. Does that kind of characterize the value of this paper, that it does at least try to do the calculations for us?
Yeah, and we can start to go into the details of what the assumptions are and what the measurements and how he came up with this formula to get to that. But for those of you that just want a quick summary, you can tell from the abstract here that if it's postulated that you need a minimum of 40 and probably more like 100, somewhere between 40 and 100 nucleotides in the correct sequence that has a self-replicating activity. That's a key obstacle in the origin of life research. That you can get some kind of molecule that can self-replicate or help replicate another molecule, that when they work together, of course, the more molecules you have, the more your probability goes down, because then you got to get the right sequence for one molecule, and you also have to get the right sequence for another molecule, and then they have to find each other somewhere in this, hydrothermal pool or oceanic geyser or something, and stay stable long enough so that they can work with each other. And he's realizing, he says, the L minimum must be shorter than 20 nucleotides for the abiogenesis probability, close to unity on a terrestrial planet. However, so in other words, the odds may be possible if it's shorter than 20 nucleotides, but 20 nucleotides seems to be too short to have any kind of self-replicating activity. So the solution is, let's hope that there's more than 10 to the 22 stars, or maybe more than one universe to make this possible. And again, I don't think it's that. We'll talk about why that's not even going to help at the end.
And at the very last, that's what I had to read this carefully. And if I totally butcher his intended meaning, is that he said, therefore, if extraterrestrial organisms of a different origin from those on Earth are discovered in the future, it would imply an unknown mechanism at work to polymerize nucleotides much faster than random statistical processes. I got hung up on that sentence, but it's also, it's like, well, you could also invoke that for even our present life, that there's an unknown mechanism, at least one we can't discover in the lab. But that echoes something Steve Benner said, who was quoted a lot in Stairway to Life. Steve Benner's, he wrote this paper in 2012 on all the paradoxes of life. I'm just like, you articulated the case against abiogenesis better than anyone I've read. So why do you believe it? And there's like one sentence he says, well, there could be new physical laws in work that we haven't discovered yet. And I'm like, okay, I see what your out is, but that's pretty much all you have at this point, because he said the asphalt paradox says structure theory predicts if we have organic systems, they're going to become useless molecules, at least as far as the life, the sort of life we're trying to solve. And so at the very end,
he's putting words in their mouth to say, because I do not want to invoke an intelligent being, I'd rather hold out for something that hasn't been discovered yet.
Yes. I mean, he's really saying either new laws of chemistry or physics. Right. I'd have to reread the paper, but I'm just like, it would have to be on that order. But I could also argue, okay, what characteristic would that new law have to be, quote unquote? I'm like, well, it would have to be able to make things that look like you need intelligent design. So you might be able to invoke something that is not personal, but it would have a similar skill set to God. So anyway, so what I could do, because we're going up on an hour and 45 minutes, we could just read your highlights here.
Sure.
Said, however, a more fundamental and unsolved problem is how an RNA polymer long enough to have a self-replicating RNA polymerase activity, i.e. RNA replicase ribozyme, emerged from prebiotic conditions and then triggered Darwinian evolution. So
that's key here. The biological or Darwinian evolution cannot take place until you have replication. So there's a whole lot that has to happen before you get to that point. And before biological or Darwinian evolution, in other words, before replication, there's the chemical and physical selection process, which is totally different and doesn't draw you towards life. It just results in molecules interacting that are going to be the most stable, or they're going to link together based on their concentration or reaction temperatures and conditions, but those have nothing to do with whether it's going to form something that will be useful to life. In fact, oftentimes it'll take you further away from life. It'll end up making aggregates and things that are impossible for life to use.
All right. And so there is actually one thing that you pointed out. You said in chemistry, there's you can't invoke natural selection. It's it's what ends, your reactions are predicted by physics and chemistry, not any sort of selection in the Darwinian sense. And I've tried to point out what people call natural selection is really just their fantasy, and they're giving it the label of natural because it's actually unnatural. What would have to be selected is unnatural based on our understanding of chemistry and physics, and also experimental evidence would show that, like the Spiegelman monster, for example, showed shows what really naturally happens. The key quantity is the minimum RNA length required to show a self-replicating ability. RNA molecules shorter than 25 nucleotides do not show a specified function, but there is a reasonable hope to find a functioning replicase ribozyme longer than 40 to 60 nucleotides.
Hope. Got to have that hope.
Hope. RNA.
Funny word to see in a science paper.
Yes. RNA polymerase ribozymes produced by in vitro experiments so far have a length longer than 100 nucleotides. And you mentioned 100. So, 60s, 40 to 60 is a hope. 100 is at least with experimental evidence. Furthermore, formation of just a single long strand may not be sufficient to initiate an abiogenesis event. No kidding. Instead, a pair of identical strands may be necessary if one serves as a replicase ribozyme and the other as a template. Yes, you need more. Your problem just got squared.
That's right.
Polymerization of RNA in water is a thermodynamically uphill process, and hence reacting monitors needs to be activated. Oh yes,
that's what we talked about before, where it's a lot of times you need the addition of phosphate groups or something that's going to impart some more potential energy to load up that molecule. In other words, you're kind of charging up that molecule so that it will overcome some of the energetically unfavorable reactions. And of course, they become unstable once you've activated the molecule as well. It adds to their instability.
and as James Tour said, where are you going to get the imidazole activated ribonucleotides?
Yeah.
Because I started to look at the synthesis experiments. And again, I'm not a chemist, but I was just like, oh my goodness, this is a nightmare. To even get you needed the raw ingredients to set it up, and then you have to carry it out with all the, that as you're building it, and it's illustrated also in Stairway to Life. You're starting a reaction, stopping a reaction, cleaning stuff up, resetting, and then starting a new reaction, stopping a reaction. This is like a factory process. It's not a soup at that point. And you only need to go wrong a few times and your polymer's gone. So anyway, some experiments yielded production of up to 40 mers of RNA.
We're talking about with clay, using montmorillonite clay as a catalyst.
Oh yes, so that was the episode James Tour was saying. They keep representing it as clay. He said it's not like any clay you could expect in a prebiotic. And I didn't realize, honestly, I was giving them more credit than they were due, because people say this is clay and there's lots of clay in on the earth. I didn't realize that in reality, this is all very specialized and with a very careful process. This wouldn't happen in natural environments.
Right, the clay is made up of metal ions. You know, they have to be in the ionized form, which are going to attract a bunch of gunk, and so they have to heat up the clays. And partly, to be fair, they have to destroy any other biological or organic contaminants on it. So they got to heat up the clay to, in case there's a bacteria or fungal spore or viral DNA on there or something, that they need to clean that up. But the clay catalyst, like any catalyst, needs to be use either strong acid or strong base to try to strip off some of the things that would be attached to the metals and get a nice clean. You're basically cleaning up the clay with temperature and acids and bases so that it can perform its catalytic job until it gets gunked up. And like Ryan Hayes points out, because he works with catalysts, you can only use them maybe a dozen times or so before they get all gunked up, and you got to clean them up again. Those clays just can't keep on acting like catalysts forever. They eventually lose their catalytic ability.
Yeah, that resonated with me. I'm just like, I wanted to hear more about that. So this would apply with these clays. They would get gunk. Wow. Okay. All right. So an experiment. All right. So, however, these results have not been reproducible. Some experiments. So it talked about some experiments yielded production up to 40 MERS of RNA. However, these results have not been reproducible.
Remember the three major tenets of science, the observable, repeatable, testable, repeatable. And so it's not good when other people can't reproduce your research.
This is really bad. You can't claim that it actually convincingly claimed that this will work, because it could be some quirk that allowed them to get the RNA, and it's not really, at least the way it was even described. Wow, I didn't know it was this bad. And only short oligomers of up to 10 MERS were produced conclusively in recent experiments, with the abundance rapidly decreasing with oligomer length.
Okay, I don't think you guys caught the significance of that. What natural selection is doing is favoring the replication of shorter and shorter molecules. Like if you're in a race to make copies of a book with someone else, and you start off with the same books, but you're like, you know what, I'm going to get this done faster if I tear out some of the pages of the book, and then you get done making copies of your books faster for the prize, and you're like, see, I'm done. And you're like, but you got rid of some useful information in the process. So the one who wins is the one who can replicate fastest. Does that natural selection favor getting a more complex, information-rich molecule, or does that kind of natural chemical, physical selection favor shorter and shorter and shorter molecules? And that's exactly what they found in these experiments.
And what I'm reading here, when they talked about 10 MERS, these weren't even replicators. This is just making a polymer, an oligomer, to be technically correct. 10 nucleotides strung together. And by the way, as we covered in the first hour, this is even an extremely generous assumption that you even could get to this stage, because they're building this with fully formed nucleotides.
That's right. Mm-hmm. Usually what they'll do is they'll add a template like this, and then they'll add the individual nucleotides, of course, in the pure, the proper isomer, and they say, hey, look, they can link to each other. The blue, the pink, and the purple here, the adenine and uracil are linking to each other, but they're not necessarily connected through the phosphodiester bonds. They might just be stuck by hydrogen bonds. Wherever this purple one finds a pink one, it'll hydrogen bond to it, but there might be a gap here, so they're not actually connected through a phosphodiester linkage here, like
a covalent bond,
like the template strand is. Right. Exactly.
Whoa.
You need like an enzyme called a ligase that's going to help form those phosphodiester linkages to make the backbone.
Oh, so this is how they would actually be able to try to.
Well, in one sense, you could say, look, I replicated the information on this strand with these, even if they're not necessarily connected to each other, or if they do somehow come together under the right conditions, you may not get an entire copy of the whole thing. You say, hey, look, I copied part of it, so I made a self-replicating molecule. I added a template, and I got a copy, and here's my replication. Like, oh, okay, great, 3 or 10 nucleotides. What are you going to do with that?
Wow, I didn't know. I have to tell. I mean, I'm glad you and James Tour are covering this, because I really felt like I was knocked to the back of my heels, so to speak, when the other side was arguing, because I didn't feel comfortable with the chemistry. But now, especially after he read it, I'm like, I feel a lot more confident that, because it's intimidating to look at these chemistry papers and you don't have a chemistry background. But when I saw James Tour, and then that show where you were with Ryan Hayes and Cy Gart, and you're all saying the same thing, I'm just like, okay, these are professional chemists. And then James Tour is saying even his synthetic chemists were saying, you're right, there's nothing fundamentally wrong in your chemical analysis of this. So I feel this has helped me feel a little more confident to say this is, I can read it and say this doesn't look good, but you're reinforcing the fact that my perception is correct. This doesn't look good.
Well, you can see how easy it is to be manipulative where the majority of people don't understand what's going on here, where someone like Dave Farina can say, hey, we've got this replication thing figured out, and without going into any of the details, any of the problems, not even mentioning that they get shorter and shorter, and that you didn't start with just these chemicals were all in the right isomers to begin with. Oftentimes a template was added to begin with, or energetic molecules were added to assist this, or that you had the right cycle of annealing and melting temperature phases. And there's all sorts of things that go into making that experiment happen that required human innovation and interference that can be swept away by just saying, hey, we've gotten molecules that self-replicate in the lab. And then everyone who doesn't know any better says, oh, problem solved. The Origin of Life researchers solved self-replicating RNA, and completely dismissing all of these issues.
Wow. And this is me even after, taking biochemistry. And it's still kind of, I said intuitively I feel that they're wrong, but I don't feel, I don't feel like I could make a rigorous argument against it. So this is very good for me. Now, I'll read on. It says here, an experimental difficulty is that aggregates may be easy. Oh, before that sentence, this trend is also consistent with theoretical expectation for random adding of monomers. So I'm saying that seems to be the theoretical expectation of 10 mers. So the 40 mer is the one they can't replicate. They can replicate 10, and that's what they said is what they expected theoretically. So it looks like...
Why is it at 10? Because if you get something that's 12, 15, 20, 30, they just spontaneously hydrolyze and break. And so you're constantly breaking them down at a rate faster than you're building them up. So by chance, you might get a few of them to link together under really good conditions using pure chemicals in the right isomer form. But the Le Chatelier's principle dictates that because it's an equilibrium reaction, you've got a lot more water than you've got nucleotides. The tendency is to break it down faster than you're going to build them up. And so that's why, at the most you'll get, 10 or 12, and then they just start breaking down. If they didn't take those molecules out for analysis and just left them in there, they would just keep breaking down until the actual nucleotides and bases start disintegrating.
So that would really
individual parts.
Yeah. So that would really cast doubt on that 40 mer claim anyway.
Yep.
So again, at every stage, it's always on such incredibly thin ice. From everything up until this paragraph, it's like your hope. Then the next sentence, an experimental difficulty is that aggregates may easily be mistaken for polymers, depending on detection methods.
You get a false positive result. You thought you got a nice strand of RNA, and it turned out to be just a clump of these that just kind of randomly stuck to each other. Or
wow, wow. Yes, RNA chemistry is difficult because I've been kind of tracking the progress of an ENCODE follow-on or related project called the E4 epitranscriptome. It's called Enabling Exploration of the Eukaryotic Epitranscriptome. It's just called E4 because it has 4 E's. And they said one of the difficult problems is analyzing RNA chemistry. It's so fragile. It's difficult to detect what's going on, much less looking for things like methyl modifications for post-transcriptional modifications. So not just the RNA, but they were saying this is really difficult to look at. So just moving on, it says, it is theoretically speculated that terminal ligation of oligomers may hierarchically produce further longer polymers, but there's no experimental or quantitative demonstration of the starting point from realistic prebiotic conditions.
So what that's saying is, rather than try to get one molecule that keeps growing longer and longer until you get at least 40, what if you had 20 nucleotide long molecules that then linked up together to form 40 and you get long polymers that way? They're saying that just doesn't seem to be the case, that you get these fragments that that then link up. And there is a certain orientation to these too. There's the 5' end, which has the phosphate attached, and then the 3' end is just the hydroxyl group on it. So we can tell the orientation. There is like a, just like when you're reading sentences on a page, you go from left to right in the English language. With these nucleotide or nucleic acids, we can tell the orientation, and we call it the 5' to 3' direction. So the 5' end terminates with a phosphate group, and the 3' end terminates with a hydroxyl group, and that's how we can tell the orientation. But that's just because of the way they're synthesized with sophisticated polymerases, DNA and RNA polymerases.
Well, I think the bottom line is that I would have presumed that they would have been able to, in their experimental setup, if there was this the speculation of the 20 MERS linking up, we would have probably seen it in the beaker, so to speak.
Right.
Okay, so I'll just go on. Just out of curiosity, it says here, a report of experimental production of long polymers greater than 120 nucleotides by ligation has been subject to reproducibility and the aggregate polymer discrimination problem. That's not the most clearly stated, but
right.
It seems
they were able to link them together by probably using the enzyme ligase. So you get fragments that then an enzyme will add the will create that phosphodiester linkage, but
it's saying
it can't reproduce it, and there's the problem is, what is it, truly a polymer, or was it just like some aggregate? Like, if it's sometimes hard to tell in the machine whether they're, I don't know if you can see that, maybe zoom in, if they're sort of overlapping, if they're kind of overlapping like this, or if they're actually connected this way, because if they're connected even like this, that's not truly a ribonucleic acid. That's just something that linked to one of those extra hydroxyl groups sticking out that was in the wrong position. How can it tell the difference between an OH that's above it and one that's on the end of it to continue the chain?
Oh my goodness. I didn't know that it was fraught with these many problems. And that's why Dr. Tour is saying the whole problem of characterization, that's another challenge. So this is painful to read through, because like at every sentence,
there's a caveat.
Like, this may not work, but we hope. goodness. Okay. I'm sorry. I was just, the reason that people will call me a nerd is I just couldn't stop reading. I just found that so compelling. All right. This is just riveting. This is just riveting reading for me. So, by the way, thank you for suggesting we do this, because you're teaching me a lot of the chemistry. It's like I can kind of read it and think that's what it means, but you're reassuring me that I'm reading it correctly. So, a high concentration of oligomers is necessary for ligation to work efficiently, but this may be difficult because oligomer abundance rapidly decreases with oligomer length in polymerization by monomers, even if such ligase activity exists. So this is maybe again like to get more concentration. How do you get more chemicals? How do you get more of that molecule in one place? I mean, it could get diluted. Any number of things could dilute it.
Well, just in building this molecule, I'm eventually going to run out of my kids' parts the longer I get. So unless you have a way of creating more parts quickly in the area that this happens to be growing, what it's saying is you got to have a high concentration, so the chances of getting them to hit each other at the right velocity and the right angle and have the right temperature and conditions for it to grow by one more nucleotide. Things have to be just right for that to happen. But every time I take one out of solution and stick it on here, now I have one less in solution. So I've got to have a way to create more of these parts, or this thing is going to be sitting here like a fragile, frail little strand, just hoping that ultraviolet light doesn't zap it, or that the side chains don't become chemically reacted, or that water isn't going to split it here or cut off the end pieces. And it's like, hurry up and make some more pieces before this thing falls apart. We've got to get it to fold in the right shape and do something functional, or we got to go find another one of these somewhere else so we can interact with it. And we're running out of building blocks here.
Right. So what do you call those things again on your kid's toy?
They're called bunchums.
So if like a bunchum is like 3 miles away, it's not gonna do any good. You have the improbability that bunchum's gonna bump into another bunchum.
Yeah.
If
you've
far
got
away.
like two or three of them in your whole swimming pool, and you're like, what are the chances that this is going to fly by and happen to interact with that one or two that are in the giant pool?
and so are we still talking about polymerization by this mononitrolic clay?
That is going to help hold this molecule onto the clay surface, and it's going to help weaken the bonds so that these two will start interacting with each other. So if my hand is the clay, because the phosphates on here are usually ionized and negatively charged, they're going to bind to the metals in the clay that are positively charged, and it'll hold it onto the surface long enough to, for one thing, to hold it in place, and the other to help weaken the bonds that are going to allow new bonds to form in between here. And then it's got to be released from the clay, which is going to require a high enough temperature to vibrate this off and break those ionic interactions so that it can go and fold into its three-dimensional shape or make a copy of itself. It's not going to make a copy of itself if it's sitting there stuck to clay. It's got to be moving around where it can fold into something, and then it needs to be, at least the way polymers are copied today, they're linear, because there's machines that will feed it through an opening and out will come a copy. Monomers will enter the machine and be linked together, and you'll end up getting two strands from this one as they're linked, as they match up and then chemically linked to each other. So they're held to this parent strand weakly by hydrogen bonding, and they're linked to each other by covalent bonding, which is much stronger. And then once that happens, they can come off and form another chain. But again, when they're dried out or when they're stuck on clay, you've really limited the possibilities for like folding that molecule or getting it to replicate or participate in other reactions with other molecules.
So I seem to remember that in Dr. Tour's series. He said, you have to get it to release from the clay. Oh, goodness. Again, another sentence on thin ice. So not only is it all these things that we're identifying, all the fine print in between that we never read.
And sometimes some of those metals are not the kind of metals you want in life. I just researched something called leadzymes, and it's where an RNA will bind to a large lead ion, a metal ion, which is positively charged. And then again, those phosphates plus the slightly negative charges on the oxygens and nitrogens and the sugar and bases will attract a lead, and it folds the RNA in such a way that it creates a ribozyme that's destructive. They think that might be one of the ways that lead causes toxicity in our cells. And we know it destroys proteins by disrupting disulfide bonds through the cysteine-cysteine interactions, but it also could be destroying our nucleic acids by forming these leadzymes.
Wow.
So,
well, just briefly, people are asking, they're really impressed, and they're asking about your channel. Does anyone have a link to James's channel?
so far, all I've been doing is supporting other people's channels. Now that I'll be done with giving a final exam on Tuesday, and then I'm thinking of creating a channel. I just don't want to compete with all my YouTube friends as it is, but maybe I'll have enough content that it'll be synergistic with what's already out there. So I may transition into doing this more full time. Right now, I'm still trying to get caught up in the literature and look at some things that I haven't studied for years because I've been so caught up with creating courses. And so I may end up creating a channel this summer, but I have to think about it some more. So right now I don't have a channel. I mean, it's just got like some home videos on it and stuff, nothing creation science related.
Yeah. Well, whatever you decide, and I'm here to work with you, because I will never view you as a competitor, because just frankly, the business model that I'm adopting is to try to just throw stuff up on YouTube. I will tell you that when Emery and I had some discussions, I said, Emery, I think I'm losing viewers because I'm doing all these nerd sessions. It's just too much for them. I began to see my subscriber numbers keep going down. It's like, yeah, I think we just kind of scared some people off. So I just needed to point that out.
Well, you have a lot of variety too. Like I've really been enjoying the various topics that you've been having, from a religious, biblical perspective as well. And I think I think we do need to cover that gamut of different approaches for, bringing favorability and light to Christianity. So yeah, and we're always careful to not step on each other's toes in terms of not broadcasting at the same time. But we don't always know when someone's going to do an impromptu session, and someone can just watch it at a different time. But I think we do need to have these technical sessions, because if we just say, abiogenesis can't work, and then it's an argument from authority, and it's like you really don't understand why until you get into the chemistry of it and the probability of it. And so for those that are interested, maybe chemistry was scary for them, but this gives them a reason to go back and learn it. It's like, oh, if I can understand this, it has practical applications. I don't just need to understand what orbitals are and what covalent and ionic bond are. I can see practical application here. I can start to understand why RNA is so fragile, and how protein folding makes a molecule work and interact with other molecules. And so I think it's necessary. Those that are overwhelmed by it, they're just going to watch some of the lighter stuff or the different stuff. And those that want to be able to learn these arguments well enough to maybe even teach them to others, they have these channels with this kind of heavy, nerdy content in it.
Well, I just this conversation I had with Rob Sadler too, and a lot of us are thinking, how do we get people interested? And that's what I was saying, is that God has to put it on people's hearts, because this is not easy learning. This is, and what I was saying, my business model is that I'm going to go with Sandy Pigeon on kind of a tour in the DC metropolitan area, face to face, in person, and say, okay, where are you? And maybe I'm going to say, okay, this is a la carte. This is like a buffet. This is too nerdy for you. We have this for you. That's how I expect the channel to grow. It's not, because the Google search engines are not going to help us very much right now. People are on YouTube for entertainment. And, I'm going to try to get some people to, that's why Sandy and I were having talks about Christian tracks. I'm going to actually try to build a website, and they go to the website and it'll be more organized. So one problem with YouTube is it's not very well organized. Now, if you make videos, I can link to your videos from my website. So that's how I can help grow your channel. But it also helps my website, because the role of the website is not to just do my content. It's organized, and it's not my content. What has happened, just for the viewers' notice, if I find a really good video and I say people need to watch it, I can't just upload it to my channel without getting some complications. Sometimes it'll get blocked and say, this is content from someone else. It has a copyright. It has copyright issues, and YouTube will send me a message and say, well, no, we're not going to, we can't allow that video to be aired on your channel. And that's fine. At least they tell me, rather than just shut down my channel, they said, okay, your channel's okay, but you just can't do that video there. And there's some things from some of these animators that are just great animations, and I can't actually show it. So the way that I actually find out whether it's there's any hold on it is I actually show it, and then I get a warning later. They don't shut your channel down unless it's someone that really wants to just ruin your channel. The policy is that either they'll say, okay, you can't monetize it. In a lot of cases, half the time they say, we'll just give the money to someone else. You show it on your channel, but when there's an ad on your channel, we'll give that money to the original owner, which I think is actually better than just shutting you down. And then, like in a case I was showing the Eric Benner thing when he was spinning the dishes.
Yeah.
They said, no, you're blocked. You can't show the entire video. You can trim it.
That guy's probably dead. Like, he's not worrying about collecting royalties from a...
Yeah,
it was an Ed Sullivan show, and
yeah, it was the Ed. You did see the Ed Sullivan. So,
well, with the thing with this heavy stuff, just there's been studies and through teaching experience, after about 2 hours, especially when you're doing heavy stuff, even the sharpest, most patient minds that aren't ADD have trouble processing. Okay, so it's good that you're breaking up in chunks. I think we need to do that with this paper as well.
Okay, so
like
we
you did with Emory. Yeah, because there's so much to
part two. Okay,
yeah, for sure.
By the way,
and for those that want the really simplified, more entertaining version, those of you that have the attention spans of a goldfish, we are trying to reach that angle as well. This team of scientists and I that meet every week on Zoom. We're almost done with our first animation, but we're trying to keep our animations less than 10 minutes and to try to explain the problems with abiogenesis in more of a simplified, even humorous way that covers the material and quote and references the appropriate literature, but breaks it down in a way that's memorable and
yeah. Great outreach.
Even kind of entertaining. So we're going to be done with our first video soon, and then we have at least five more planned that we'll probably get 3 more done just in the summer alone, because a lot of us won't be teaching in the summer. But it's taken months to get through the first one, and we're also doing this through the Discovery Institute, so we need to get their blessing on it and have it go out in a strategic way so that we can try to make it go as viral as possible. So that would be something that people can help with, is like, hey, yeah, here's a short video that shows the problems with abiogenesis, and you can post it on your social media or whatever you can do to help that get out, and then that will get people interested. And if they want to go dig into the details, they can read. We put the references in there. We put a book by Dr. Tour and also Change Tan, Rob Stadler. We give links to their books, as well as we could do talks like this where they can go into a deeper dive into the details.
Well, I wanted to say, even if people don't watch the entire video, it makes a statement. It's like, okay, they might superficially say, some of the critics, like, you
didn't cover this.
But then it's like, no, the people that helped author this, like yourself, the video, they will go into the painful details. And if you're really trying to be fair-minded, you're going to watch.
If you're eager and you're willing to be open-minded, we're willing to teach you the details.
So here's, Journal Club is becoming a favorite theme. I get so much more out of a paper hearing professional level commentary. Yes,
I've been enjoying these. Yeah.
That's why when Dr. Carter offered to he said, why don't we talk about this? I said, yes, because you can help all of us. Because, we may actually understand parts of it, but we're not sure that we're understanding it right. Because if I were reading this, I said, it can't be this good news. I mean, which is really bad news for them. But each time they say this is problematic, that's good news for us. And then as I read between the lines too, it's like, it can't be that bad for them and that good for us. And it turns out my impression is generally correct from what you're telling me.
Yeah, it shows we're not quote mining or taking things out of context too when we go through this piece by piece. And
so we can resume this another time, because I'm also running out of gas myself. And you did tell Mrs. Carter that this is only go on for an hour. So
I'm going to have to massage her feet now. Thanks a lot.
Yeah. Well, so I thank
Andrew
you very much for your time. Thank Mrs. Carter and your family.
Oh yeah, sure. Yeah. It's wondering when the animations will come out. Probably through the Discovery Institute. Actually, it's going to be on Long Story Short. He's the animator, Evan Diaz, who's doing the animations for us. So he's got a channel called Long Story Short, and that it will be advertised and promoted through the Discovery Institute, as well as we've set aside some money for advertising through like the 70 Adventist churches and any other church that wants to promote it somehow, because it is at a layperson's level. Then we just want this to get out to as many people as possible, so that it is crucial that it happens in the first week after it's released too, because of the algorithms to try to get it to go more viral, especially on things like Facebook, where it's like, if you don't make comments on a video right away in the first couple hours, it just gets lost into the no man's land of
search engines.
Yeah, exactly. So that's what we need help with, is just trying to amplify it by getting as many people to post it on their social media and make comments on it. And then, because we're going to have a series of these, you can, subscribe and get notified when we get finished with the next one. So, that's for those of you who need the nice, pretty colors and animations and sounds, and want to be able to share something with someone who doesn't want to go into the painful chemical details.
Well, I will tell you this, even for someone that likes going into the painful chemical details, I got a sneak preview through Rob, and Rob said, don't share it. And
Good.
I haven't,
yeah.
But I'll just tell you, it's good. So even someone that loves the details will say this crystallizes really everything that we talk about flows from the simple stuff that was in that cartoon. And you can frame everything that we've discussed here in terms of that cartoon. We're just giving it more fine detail, but it's essentially the same argument. We're just, yeah. All righty.
Well, sure good to do this with you, Sal. And
likewise.
Nice to see you smiling. I know it's been such a hard time for you lately, and missing your mom in the background.
Oh, goodness. I mean, if I am genuinely in a good mood, but if you saw me this afternoon, I was with my sister. I was bawling my eyes out. Part of the reason I'm in a good mood at night,
cathartic.
Is like, I'm gonna cry all during the day, and then it'll put me in a good mood. But I am comforted. She's in a better place, and I thank you, Dr. Carter, when you're sending me messages on my phone and encouraging me. I want to let the viewers know what a great guy he is, and helping me along, and Emery during this time, and all my friends. The outpouring of love and sympathy and support has been just overwhelming.
Yeah, and we're sure proud of you, and we're praying for you. And even if you did, I know it's got to be really strange. I was thinking, like, you spent so many years being a caretaker, and suddenly this huge chunk of your love and your time and resources have just been freed up, and how lonely that must feel, just being in the same environment. So if you want to even do these chats on or off air, where we can just lift each other up and support each other, I'm all for that. We don't have to always talk about heavy science. These things matter too.
Well, this is talking chemistry is also a form of fellowship with me, because it's how I can see the wonders of God. And the live shows are very comforting, because it's just nice to see people that want to listen to what I have to say. And that means, I can't tell you how much that means a lot, how much that means to me, that we have this community where people will, put me on their YouTube.
Yeah.
And I have a chance to share. And, Mom would want me to carry on with this work. And so she would be happy that I'm doing this, and she wouldn't want me to be grieving about her, because Lord willing, I'm expecting to see her again. This is only what the priest said. She's Catholic, so she had a Catholic funeral, but what the priest said, he quoted Thessalonians, that we do not grieve as those who have no hope. This is only sleep. That means I'll see her again.
That's right.
And this separation is only temporary. And I said, that's what makes what we're doing here all the more important, because it reassures us this isn't a fable or a myth. The one who can, if there is a creator God that can make the miracle of life, he can also make a resurrection. This is our assurance that this can happen. So, this is, I can't tell you how much comfort this is. And yes, God bless you all. And see, we're reaching out to people. And again, I hope we'll be able to serve all levels of knowledge. So this is like really down into the weeds, to use landscaper terminology, down into the weeds, down into the roots of the weeds at this point. But I do hope to have, with the cartoons the animations that are coming out, they're only 10 minutes, that Dr. Carter and Dr. Stadler, Dr. Hayes, and have been working on for months to give 10 minutes. That'll be just, I'll happily share it on my channel and on my website. This is going to be just glorious. And so this coming week will be kind of busy. I have my debate. So maybe if you want to try to do this again next Sunday, we can try, now that your semester's. So whatever your schedule works, you can email me, Dr. Carter. I'd be thrilled.
I'll keep in contact. It's a 4-day weekend for my son, because he's got Friday and off, and then Monday is the holiday. So we may end up going somewhere, but just let me know when you get back from your trip, and we'll do part 2.
Oh, okay. Yeah, we'll follow up, and maybe the video will be released by then. So before I say goodbye, I'd like to thank all the viewers here who've joined us at the Evidence and Reasons for the Christian Faith video channel. The purpose of all this is not ultimately to just nerd people out. It is to bring you all, the believers, closer to God, to reassure you in your faith and strengthen you in your walk with him. So for all those who actually went through this grueling session, I'm praying and hoping that the Lord will comfort you through this process. So Dr. Carter, I'll give you the last word, and then we'll shut it down.
Yeah, I want to thank you guys for bearing with us through some pretty technical stuff here. I know sometimes I have to listen to a video twice just to get all the details, or I'm half paying attention to the comments and chat, and then half listening to the video. I'm like, hey, this was something that was important enough. I need to go back and listen to it a second or third time to make sure I capture all the information. So it's I think if you're going to go through these challenging topics, it's better if you not distract yourself as much in the chat. I know I'm guilty of that, and I find myself just diluting my attention, just like if a student was texting while I'm giving a lecture. It's sort of, you might get a little bit of short-term memory, like, oh yeah, and then you're like, what's going on again? So we'll try to do little recaps here and there, say, okay, that was a bunch of heavy stuff, so now we're going to step back and look at the forest for the trees and sort of tell you why this matters, why this is important, and then go on to the next one. So we've covered plenty in this session. I'm sure we'll have at least a part two. We'll try to be more succinct and focused on the rest of the paper. But I want to thank you for all the encouragement, and the summer is going to be exciting. I'm going to be on YouTube a lot more now that I'm done with teaching. I'm considering even sort of retiring from teaching early, just so I can do more stuff like this full time. If God can provide a way to do that and provide for my family, which I trust He will. But I just think this is so important that we reach as many people as we can with this crucial information that's faith building, and it's just beautiful too. It makes you just respect and bow down in awe of our Creator, of the foresight and planning and engineering that goes into making life happen and making it sustainable so that it can continue to go on once that clock is wound up. It's just there's so many times where I'm studying new things or trying to teach things that it just absolutely blows me away. I'm like, who can I tell about this? This is just incredible. And if only people could see what was really going on at the molecular biochemical level, they would have such an appreciation for God and a understanding of the limits of natural processes and natural selection. So I want to encourage you guys to be brave when you come out in public like this. It can affect your career. Fortunately, I'm at a Christian university, an accredited university, but I think that there will be someday, because this is a university that's accredited by a very large accrediting body, the Western Association of Schools and Colleges, that there will be a time where they will say, hey, if you're not teaching evolution only, or if you're throwing in religion and stuff, you're not going to get accreditation, or you're not going to get bonds to build new buildings, or I think that there's a spiritual war going on that we need to be cognizant of, and you will face persecution. It's challenging, especially those of you that are introverts or have anxiety issues, or you're stepping on the battleground when you start sharing this kind of information with others. And if we all have guardian angels and a third of the angels were fallen, that means there's billions of powerful demons that are working to try to trip you up and prevent you from hearing this information or understanding this information, or being distracted, or being too shy to share it with someone, or getting so caught up in the details that you don't make a decision about something because you're like, well, I see arguments on both sides, and so I'm just going to keep waiting on the fence, because there's going to be a time where a decision needs to be made, and we can't just stay in limbo forever. These have very real implications for the future and the way that we conduct our lives. So it's important that we devote enough time and prayer and attention, and we need the power of the Holy Spirit to help give us wisdom and discernment. So keep praying for that. If any of you lacks wisdom, and pray, and God will give you the wisdom and the ability to discern these things.
All righty. Thank you, Dr. Carter. Lord willing, we will meet again. And thank you, all the viewers. Have a good night.
Sal.
And God bless you all. Thank you, Dr. Carter.